GEMSA staining. One of the most common staining method of the chromosomes is called as GEMSA staining. GEMSA staining usually gives a stripped appearance to the chromosomes, in which we can see the dark bands and the light bands. They are repeated. Some of the bands they appear as dark, and some of the bands they appear as light in color. So the dark bands are those bands they are rich in adenine and thymine. So there is a rich dark band appear as rich in color of the darkness because there is presence of adenine and thymine in those regions. And GEMSA stain that bind to AT rich regions, that is the reason these bands they look dark in color. In this diagram we can see that how a chromosome appears when we stain the chromosome by GEMSA stain. These are the dark bands. We can see here that this is dark band, and this band is called as GEMSA band, and this is the light band. Again here is dark band, then light band, this is dark band. So chromosome shows dark band and light band in the form of strips. So dark band are those bands where they are rich in adenine and thymine. GEMSA stain that attach to this AT rich regions, due to AT rich region chromosome shows dark bands. Dark G bands are those bands that replicate late during the cell cycle. As we know, while dividing the cell into two cells, the DNA or the genetic material of the cell divides and it replicates. So those DNA which is present in the dark bands, that DNA replicates late during the cell cycle, and this DNA is AT rich region. While on the other hand, light G bands they replicate early during the cell cycle. So this is the difference between two types of the bands, dark G bands and light G bands. Dark G bands are those bands which are AT rich and they replicate late during the synthesis in S phase of the cell cycle. While on the other hand, light band are those bands which are GC rich and they replicate early during the synthesis of the DNA during the cell cycle.